Hey guys, welcome back to our channel. So this is the part 3 of Cell the Unit of Life, and let's get started. Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and functions, many of these are considered together as an endomembrane system because their functions are coordinated. The endomembrane system include endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. Since the functions of the mitochondria, chloroplast, and peroxisome are not coordinated with the above component, these are not considered as part of the endomembrane system. The endoplasmic reticulum electron microscopic studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm that is called the endoplasmic reticulum. Endoplasmic reticulum divides the intracellular space into two distinct compartments: luminal inside endoplasmic reticulum and extraluminal cytoplasm compartments. The endoplasmic reticulum often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosome on their surface is called rough endoplasmic reticulum. Absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum. Rough endoplasmic reticulum is frequently observed in the cell's activity involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is the major site for synthesis of lipid in animal cells. Lipid like steroidal hormones are synthesized in smooth endoplasmic reticulum. Golgi apparatus. Camillo Golgi, 1898, first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat, disc-shaped sacs or cistern of 0.5 micrometer to 1.0 micrometers diameter. These are staked parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase and concave trans or the maturing phase. The cis and the trans phases of the organelle are entirely different but interconnected. The Golgi apparatus principally perform the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Material to be packaged in the form of vesicle from the endoplasmic reticulum fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remain in close association with the endoplasmic reticulum. A number of protein synthesized by ribosome on the endoplasmic reticulum are modified in the sternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. Lysosome. These are membrane-bound vesicle structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicle have been found to be very rich in almost all types of hydrolytic enzymes: hydrolysis, lipase, protease, carbohydrates, optically active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acid. Vacuoles. The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory products, and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport of a number of ions and other materials against concentration gradients into the vacuoles. Hence, their concentration is higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and secretion in many cells. As in proteases, food vacuoles are formed by engulfing the food particles. Mitochondria, sing mitochondrion, unless specifically strained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size, also considerable degree of variability is observed. Typically, it is sausage-shaped or cylindrical, having a diameter of 0.2 to 1.0 micrometer, average 0.5 micrometer, and length 1.0 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms a number of infolding called the cristae. Sing crista. Towards the matrix, the cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the site of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouse of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecule, ribosome, 70S, and the component required for the synthesis of protein. The mitochondria divide by fission. Plasmids. Plasmids are found in all plant cells and in euglenoids. These are easily observed under the microscope. As they are large, they bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plasmids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll and carotenoids pigments, which are responsible for trapping light energy, essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophylls, and others are present. This gives the part of the plant and yellow, orange, or red color. The leucoplasts are the colorless plasmids of varied shapes and size with stored nutrients. Amyloplast store carbohydrate, starch, example potato. Elioplast store oil and fats, whereas the leucoplast store protein. Where majority of the leucoplast of the green plants are found in the mesophyll cells of leaf. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length, 5 to 10 micrometer, and width 2 to 4 micrometer. The number varies from 1 per cell of the chlamydomonas, a green algae, to 20 to 40 per cell in the mesophils. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, in the inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organelles, flattened membranous sacs called the thylakoids, are present in the stroma. Thylakoids are arranged in stacks like the piles of coins called grana. Singular, granulum, or the integral thylakoids. In addition, there are flat membranous tubules called the stroma lamina, connecting the thylakoids of the different grana. The membrane of the thylakoids encloses a space called a lumen. The stroma of the chloroplast contain enzyme required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller, 70s, than the cytoplasmic ribosomes, 80s. Ribosome are the granule structures first observed under the electron microscope as dense particles by George Palade, 1953. They are composed of ribonucleic acid (RNA) and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80s, while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, large and smaller subunits. The two subunits of 80s ribosomes are 60s and 40s, while that 70s ribosomes are 50s and 30s. Here, S. Schwarzbard's until stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70s and 80s ribosomes are composed of two subunits. Cytoskeleton, an elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagella. Cilia sing cilium and flagella sing flagellum are hair-like outer growth of the cell membrane. Cilia are small structures which work like oars, causing the movement of the cell or the surrounding fluid. Flagella causing the movement of either the cell or the surrounding fluid. They are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of a cilium or the flagellum show that they are covered with plasma membrane. Their core, called the exosome, possesses a number of microtubules running parallel to the long axis. The exosome usually has 9 doublets of radically arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of exosomal microtubules is referred to as the 9 to 2 array. The central tubules are connected by bridges and is also enclosed by a central sheet which is connected to one of the tubules of each peripheral triplets by a radical spoke. Thus, there are 9 radical spokes. The peripheral triplets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole like structure called the basal body. Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibrils is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub. This is connected with tubules of the peripheral triplet by radical spokes made of proteins. The centriole form the basal body of cilia or flagella, and spindle fiber that give rise to spindle apparatus during cell division in animal cells. Nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. The interface nucleus of a cell when it is not dividing has highly extended and elaborate nucleoprotein fibers called chromatin. Nuclear matrix and one or more spherical bodies called nuclei. Seeing nucleus electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuously with the endoplasmic reticulum and also bears ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear pores are the passes through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variation in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus. Example, erythrocytes, many mammals, and sieves tube cells of vascular plants. Would you consider these cells as living? The nuclear matrix or the nuclear plasm contain nucleus and chromatin. The nucleoli are spherical structure present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Larger and more nuclei are present in cell actively carrying out protein synthesis. You may recall that the interface nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stage of cell division, cell shows structured chromosomes in place of the nucleus. Chromatin contains DNA and some basic proteins called histone. Some non-histone proteins and also RNA. A single human cell has approximately 2 meter long thread of DNA distributed among these 23 pairs chromosome. You will study the detail of DNA packaging in the form of a chromosome in class 12. Every chromosome visible only in dividing cells essentially has a primary constriction or the centromere on the sides of which disc-shaped structure called kinetochores are present. Centromeres hold two chromatids of a chromosome based on the position of the centromere. The chromosome can be classified into four types. The metacentric chromosome has middle centromere forming two equal arms of the chromosome. The submetacentric chromosome has a centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of acrocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constriction at a constant location. This gives the appearance of a small fragments called the satellites. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both. This chapter reading is completed, and thanks for watching.